The funny thing is, what I was saying was I had problems with my mic last week. Okay, you should hear me now. Let me know if that's good. Yeah, I had muted my mic to avoid... Oh, is my head being cut off? That's fun. I had muted my mic to avoid y'all listening to me sip water and eat an apple while I had that slide on. So thank you for the ping in the chat. We had some problems last week where the mic was really loud when I was using my face and then really quiet when I was using VSCode. That shouldn't be a problem today, so fingers crossed. Welcome everyone. My name is Sarah Guthals and I'm here coming to you from the Reactor team. Reactors are physical locations around the world where Microsoft promotes communities of professional developers. So if you're a professional developer who wants to continue learning, learn something new, maybe you've never done data science before, but you've heard the term and you want to try it out, we provide content and courses and workshops and tech talks and things like that, free of charge. Typically in a physical environment, but in light of recent events, we're going digital. So we are excited because going digital was always part of our plan in addition to our physical locations. So we get to kickstart that plan. We've been doing this for almost 2 months now, streaming on Twitch. On Tuesdays, we do about an hour where we introduce a new topic. And then Thursday, I'll be doing 2 hours where I dive deeper and do some more exploration. After the stream is over, we always post these videos up on YouTube. So if you don't get a chance to stick around for the entire hour today, or you're missing it, which then I wouldn't be talking to you at this moment, you would actually already be listening to it on YouTube, but you could watch it on YouTube after the fact. Also, everything that I'm doing here today will be available on our GitHub, and so we'll link all of those things. And I also wanted to mention that we have some of my teammates, or at least one of my teammates, in the chat. So Megan today, I know, is in the chat with us. And if you have any questions and I'm not getting to that, feel free to ask both of us in the chat and one of us will get to it. The code is at the github.com/Microsoft/reactors. There's an online folder and in it a genomics folder where all of the code we'll be writing today, or even more than what we'll be writing today, exists. So you can check that out. You can do so now and kind of like follow along with what we're doing, or you can do so afterwards. For today's stream, I'm going to be using Visual Studio Code with the Jupyter Notebook and Python extensions enabled. So I have already downloaded VSCode. That works on all platforms. I have enabled the Python extension, and I have opened up a file that is .ipynb, so a Jupyter Notebook file, which prompted me to to install the Jupyter Notebook extension as well, or package. So if you want to get started, I'm going to do like a quick introduction for the next 5 or so minutes, so you have some time to get VSCode, install the Python extension, and make sure that you've got a Jupyter Notebook file open. In addition to that, you'll probably need a couple of other things. I think we're going to be using Pandas and NumPy, and there's one other package I think you'll need to install. I think it's just matplotlib. Yeah, so Pandas, NumPy, and matplotlib. So you'll need to make sure that those are all installed as well. Awesome. All right, so today we're going to talk about genomics. I am not a genomics scientist. I don't really know much about genomics other than they're important and they involve a lot of data. The reason I wanted to talk on this subject, though, is my partner is actually and I just forgot the word, but he's a bioinformatics scientist, and there it is, and he focuses on proteomics. So it's different than genomics, but same type of thing. Basically, in biological sciences, we have data that represents biological structures. You might remember this from maybe like a school-age biology, but your gene. So DNA, for example, is split up on, what are the 4 letters? A, C, D, D. See, now I'm forgetting already. I am not a scientist. A, T, G, C, right? So adenine, and excuse me, totally showing how much I don't know about this. Thymine, guanine, and cytosine. Cytosine? So if you recall, it was like, the A and T and C and G, or maybe it was A and G and T and C. I don't remember. They connect up, right? And they create your DNA. And basically what genomics is at a very high level, is the study of grabbing that genome, that sequence of the base pairs, and identifying where there might be any kind of like misalignment or any kind of just kind of like differences. And that's how we study genetic like anomalies in our bodies. Very like not even 100 level explanation, because I'm not even an expert enough to give that level of explanation, like a 10 level explanation of what that is. But basically it's we take our like proteins, our DNA, all of that, and we can run it through machines like mass spectrometers, and out comes kind of the sequence of whether it's the DNA or a protein. And that sequence is not always clean. So what I mean by that is we're grabbing actual biological samples and trying to sequence them. So the sequence might not be exactly right. And we don't know everything about our human bodies. And so that makes the data not just potentially dirty or like wrong in some ways, but it also makes the data just kind of new and interesting to explore. And so when you look into biological sciences and the researchers who are doing this kind of work, it is often paired a like machine learning or data science expert with biologists. I was going to say biological scientists, but they're called biologists, with biologists. And the biologists understand kind of the collection of the data. They understand what the data means. And the machine learning experts might understand how to clean and manipulate that data so that we can gather information about it and run predictions and things like that. A bioinformatics scientist, what my partner does, is kind of like in the middle there. So they tend to understand a lot about the code, they tend to understand a lot about the biology, and they try to kind of bridge that gap. So this kind of topic is really interesting to me because it's basically just taking really messy, hard to understand, newly discovered data and trying to understand something about it so that we can make predictions and therefore create therapeutics, or just have a better understanding of how different anomalies might affect humans. So that's why I was interested in it. In the comments, I'd love to hear, both in the Twitch stream and in the YouTube comments, why you might be interested in data science and genomics or in biological sciences. Is it just because it's kind of an interesting hot topic, or is it more than that? I'm curious to know. For example, one quick other aside, one of the reasons I'd like to know is I've had asthma my whole life, and I really want to better understand that. So in another life, I actually became a bioinformatics scientist too. I didn't end up doing that, but I did marry someone who did, so that I can kind of get the inside scoop there. All right. So next, I just want to go into a quick kind of overview of why we're going to be using a biology major looking to enter data science. I love it. Why we're going to be using the tools that we're using today, because we're not choosing these tools just because, and actually none of these are Microsoft tools necessarily. We're not using these tools because we're Microsoft. We're using these tools because that's what the industry, the biological sciences, and the machine learning experts who do things on genomics use. And so I wanted to go a little bit into that. We're going to do this fairly quickly. Again, remember our stream on Thursday is more exploration of code. We will get to the code very shortly though. Okay, so let me switch over to, we're not going to do that one quite yet. Not going to do that one quite yet. Nope, sorry, JK, this guy. All right, I think I just yelled. I'm sorry about that. Okay, so actually I'm just going to tone down my mic just a little bit. I didn't realize it was hitting the red there. Okay, that should be a little bit better. Okay, so why are we thinking about the tools that we're going to use today? So first of all, Python is the language that we're going to use. And the reason why we're going to use Python is it's already broadly embraced by the scientists who do this work. The reason for that is because, well, it's kind of like a chicken and an egg, right? One of the reasons is that there exists a lot of libraries and tools that already extend Python's functionality into the biological domain. And I'll share some of those in just a minute. Also, Python is notoriously known, I think those are kind of synonyms, but for making it easy to manipulate strings of characters, which is basically what a genome sequence is or a protein sequence is. It's just basically these really, really large strings of characters where we're going to have to kind of take them apart, put them back together, and understand where there might be anomalies. And Python works with lots of existing tools. So I mentioned a couple of libraries and packages and such. So these are some of the libraries that already exist to support research in the life sciences that all are Python libraries. So BioPython, for example, is a collection of non-commercial Python tools for computational biology and bioinformatics. So as I mentioned, like computational biology and bioinformatics, there's a huge crossover there. I'm not well versed enough to know exactly the difference, but tomorrow we're actually going to be jumping on Twitter, and I'm going to be asking my husband if he can kind of answer some of these questions for us. So make sure you check out our Twitter tomorrow. So BioPython contains classes that represent biological sequences and sequence annotations, and the library can be used, can also be read and written to a variety of different formats. And so this contains just like a lot of existing biological sequences that you might be able to test against, for example. The distributed evolutionary algorithms in Python, or DEEP, is an evolutionary Python framework for rapid prototyping and testing of ideas. So basically it incorporates data structures and tools required to implement the most common evolutionary computational techniques like genetic algorithms, genetic programming, evolution strategies, particle swarm optimization, differential evolution, and estimation of distribution algorithms. So this is more for that kind of evolutionary research where you're trying to see like if something were to change in the genetic structure, what might happen, or what might have happened. I don't know how this one's pronounced. NiLearn. I'm not exactly sure. But this one is basically a module for statistical learning on neuroimaging data. So it makes really advanced machine learning models a lot easier to work with this type of data because it does a lot of pattern recognition and multivariate statistical techniques on neuroimaging data. So basically taking in kind of the images that you would take from a brain scan and kind of understanding those. PsychoPy is a package for the generation of experiments for neuroscience and experimental psychology, similar to NiLearn, I think. SciPy is a library used by scientists and analysts doing scientific computation on basically the models are optimized for linear algebra, integration, interpolation, special functions, fast Fourier transforms, signal and imaging processing, basically just like a lot of the really common tasks in science and engineering. And Scientific Python is a collection of modules for scientific computing. So it supports things like geometry, mathematical functions, statistics, etc. So these were all built on top of Python. Again, I mentioned it was kind of a chicken and the egg, partially because that's where the scientists were already. And then also the scientists continued to go towards Python because these libraries were built. Python lends itself really well to these smaller libraries and modules as a language itself. And so I think that's what really helps support that growth. We're also going to be using Jupyter Notebooks, as I mentioned. Now, we're going to be doing it inside of Jupyter Notebooks with Visual Studio Code. Sorry, we're going to run Jupyter Notebooks inside of Visual Studio Code. The reason for that is just I find it easier to have one development environment. I particularly like Visual Studio Code since it now supports Jupyter Notebooks. Great, I can use that. But it is really a standard for data scientists, whether you're using Jupyter Notebooks inside of VSCode, you're using it in some kind of cloud platform, or you have Jupyter Notebooks installed on your machine. It really is a standard for data scientists. It allows you to have executable code cells that also have the output directly in there. You can also add in commentary, and you have multimedia resources. So for example, we can see that we can have, and we'll do this during this stream, we'll have, visual charts and visual aids, images, etc., that allow us to explore our data further. We'll get into that a little bit more, but the ability to actually run one individual cell, see what happens, make one change, and rerun just that cell is really important, because you might be working towards a solution when you're exploring this really messy, complex data, and you don't want to rerun the entire notebook or the entire file. You really just want to iterate on this one cell. And once you start to get to the point where you're fully understanding what you're building, then you can create like one Python file that encapsulates everything. Similarly, Jupyter Notebooks are also just easily shared, again, especially because we have things like cloud resources for these. We can share them fairly easily. We can have it's very easy because we can have that text as well as those images of those graphs and such embedded inside of the code. It presents more of like a narrative with working code. So you could share this with someone who's also understanding of the technology or understanding of the sciences, and they can kind of walk through it in a story-like fashion rather than just kind of running a file and seeing what the output is. And then it therefore allows this conversation and collaboration to happen, because if you are a biologist and you don't know the code very well, you don't necessarily have to look at the code, but you could have a sentence that explains what the code's about to do. You could run that cell, see the output, and have a clear understanding of like what exactly is happening right there. That would allow scientists to communicate better with the coders or the machine learning experts, because they can say, well, what if we tweak this one parameter here? And they don't have to understand an entire code file or read through the comments. They can just kind of read through this more narrative. And you'll see on the GitHub repo that we do have this as more of a narrative. And so you can kind of get a feel for what we're talking about there. Awesome. All right. I'm just checking out the install Vim. Sure, you can install Vim. And yes, I like to use just, I have everything on my GitHub repo, and then I just use GitHub Desktop because it's just easier for me. But, and then I open up everything in VS Code. If you want to use Vim, power to ya. All right. It's actually what I learned on, so. All right, let's get into some of this coding now. So like I mentioned, I have VS Code open installed. I've got the Python extension enabled. So I got the Python extension from Microsoft. I made sure that I had chosen an interpreter. So I'm just using my local Python. I didn't create like a virtual environment or anything. And then I created a file called genomics.ipynb, and when I saved that file, it automatically saved it as a Jupyter Notebook and started displaying it in that way. You should also make sure that you've got Python, that you've got your kernel started with Python as well. If something goes wrong, you can do Control-Shift-P on Windows or Command-Shift-P on Mac, and you can type Python, select interpreter to start Jupyter server. And when you do that, all of your Python versions will show up, including any kind of virtual environments you might have. And you can choose the one that you want to use. All right. Yes, I actually do want to explore doing VS Live Share in a future stream, maybe on Thursday, maybe not quite so soon, but I do want to actually do Live Share on a future stream and have maybe like the first couple people who pop in do some like co-coding with me. I think that would be fun, but we're not there yet. Okay, so hopefully if you do want to follow along, you've had the time to set everything up, and now we're going to go ahead and get started. As I mentioned, the file is on GitHub, and if you do want to follow along from scratch like I'm doing, the genome.txt file, so all of our data, is also on GitHub, so you can at least just download that one file. All right, as I mentioned, we do want to import a few of our packages and modules. So we're going to import NumPy, and I just like to use that shorthand. And it just started raining with the sun, and it looks so beautiful right now. Sorry, I got a little distracted. I've got a big window in front of me. And we're going to import matplotlib, the pyplot specifically, so that we can show our pretty plots. Oops. Pyplot as plot. And I do want to show all of our plots in line, so I'm going to use this handy dandy bit of code right here, magic command. All right, so let's make sure all of that works. Great, so that seems to be running. If you're new to Jupyter Notebooks, particularly in VSCode, you can run the cells by clicking on this this play button right here. You'll notice that the cell completed running when there is a number next to it. These numbers are important because, as I mentioned earlier, you may want to rerun certain cells, and knowing the order in which cells were run when you're creating, variables and updating those is really, really important. So, okay, the next thing that we're going to do is we're going to read in our data. So I'm going to call my data genome, and I'm going to read it in using pandas. And it's just called genome. Genome.txt. Awesome. And I'm going to go ahead and print out the top of that. Okay, so we can see some information here. And if we open up the txt file, we'll see similar information here. So we've got an RSID column, a chromosome column, a position column, and a genotype column. Okay. And we can see that the first 5 that are displayed here look correct to me, except we do have this kind of like tab information here rather than it being in a data frame. So we might want to change that up because I don't want this to be one column where we've got these /t, aka like tabs in there. I want those to be 4 unique columns. So let's go ahead and change that up. And I'm just going to copy and paste this code from up here. And what I'm going to do is, instead of just reading the CSV, I want to read the CSV, and I want to specifically mention that my separator is a tab. Oops, sorry, wrong. So this is a tab separated file instead of a comma separated file. So I need to specify that. That looks much better. Now, instead of having one column with these random tabs in there, I've got 4 unique columns. And those seem to match up. AA, GG, AG. Awesome. All right. Hello. Okay, so now we've got our our data inside of a data frame. And it's, one of the reasons that we're doing this in code is because, like, if we could have potentially put this inside of an Excel doc, but I mean, it's got a lot of rows here, and that's kind of the whole point of using code. Is I want to understand large amounts of data without having to manually go through it, right? And that's really what birthed the bioinformatics field. Is the biologists and the wet lab scientists had large amounts of data that they needed to be able to synthesize. And Excel is useful, or similar types of tools, but code kind of really makes it easy for us. So let's get an understanding of what is inside of, oh my gosh, spelling. What is inside of our data frame, or inside of our data. So this is a data frame. It's got 4 columns: RSID, chromosome, position, genotype. It looks like we've got non-null values for every single row in the columns, in each column. So we can see that there are 966,977 entries, and we have non-null values, that same amount of non-null values for every single column. That's great. That means we don't have any missing data, and we can kind of proceed from there. Now, this is the point at which I would bring in a SME, a subject matter expert in biology, if we were missing data. If we were missing the chromosome for, 500 rows or something, would that still be useful for our analysis? It depends on the question, but it might mean that we would want to remove all of the rows where that data was missing. It's important to recognize that this is when you would bring in that subject matter expert to make sure that you understand. You understand how to clean your data. Sorry, I didn't finish my sentence there. You also notice that we have the type. So RSID, chromosome, and genotype are all of type object, and position is of type integer. We can see that that's likely because there's kind of letters and numbers in here for all of them. And like at the first few, it looks like chromosome and position are just numbers. But if we kind of start to scroll down, I think we will find, yep, here we go. We will find that chromosome does have some values that aren't numbers. So that's useful for us because we might have missed that. I mean, it took us scrolling nearly to the bottom to really see that that chromosome had non-integer values. So this might have been an immediate indication to that. Is there a way you can run this online without a paid Azure account? Yes. So Oh, yes. Sorry about that. There is, the GitHub version does have a /data in there. Typically, I would put my code in a data folder. This time, I just had it in the main one. So yes, make sure that you change that if that's where you have it. Yes, currently we've got Azure Notebooks. If you go to notebooks.azure.com, you should be able to sign up and run this with just a Microsoft account. So yeah, you can try that. Cool. All right. So let's continue. First, before we continue any further, again, I'm not a genomics expert. I don't know everything about it, but we can start to explore at least these 4 columns. So RSID is basically an ID for the SNP cluster. So I do have that page open right here. And let me make sure I can actually see it as well. So basically what this is the location in the genome that is known to vary between individuals. Okay? So this is essentially my understanding, and if you are a biologist, please feel free to correct me in the comments. But my understanding is it's basically a location in this very large sequence that we know is different between people. And so that's going to be a location that we want to start looking at. Okay? And so we can start to link SNPs, aka those locations, to various traits or conditions using their RSID. So someone who does have this something in this location, or does have this value essentially in this location, is likely to have this trait. And that's how a company like 23andMe can say, you probably what is one of them? You probably flush when you drink alcohol, or you probably like the taste of cilantro, for example. All right, let's go back to our code. So that's what RSID is. The chromosome is which of the 23 chromosomes, so it's 1 through 22, or X or Y, which of the 23 chromosomes that a human has, or the mitochondrial chromosome, where the SNP appears. So again, I'm not a biologist, so if you are, feel free to correct me or provide further explanation in the comments. But basically, where the SNP lives, so that location, which chromosome is that on? And that will likely have different implications. The position refers to the specific location of an SNP on the reference human genome. So on the entire genome, what is the position? So basically the location of that SNP. And then the genotype refers to the pair of nucleotides that form an SNP inherited at that chromosomal position. So basically what we're saying is this SNP is on chromosome 1 at this position, and this is basically its value. That is my understanding. Again, if you know more than me, please don't hesitate to correct me. All right. So now we're going to actually just start exploring this data a little bit. And the purpose of today's stream is really to do that, is to start exploring and visualizing this data so that we can start to come up with questions that we might want to ask. So one thing that we could do, for example, is take a look at all of the chromosomes. Chromosome, that was right. And see how many SNPs we have per chromosome. So for each chromosome, for each of the 23 chromosomes, how many SNPs do we have in our data? All right, and this is ordered by the most to the least. Now, I don't know about you, that's like awesome. I see a bunch of numbers. The difference between chromosome 2 and chromosome 17, yeah, I mean, I could do the math. I can see it's like 50 or so thousand, but it's not extremely clear to me. I'm more of a visual person. So what I might do is, instead of just viewing this as a like table, I might want to view this as a bar graph. And that gives me a clear kind of picture of what's going on here, that we can see that maybe there is some like clusters. So maybe 1 and 2 have a lot, maybe 3 and 6, not so many. And we can kind of, maybe that's interesting. Again, this is where I might bring in the SME, subject matter expert in biology, and ask them, like, is this interesting? Are these chromosomes, is this what you would expect? Are these chromosomes the ones where you would expect more SNPs? Are we missing data? So not just kind of, is this what we would expect, assuming that the data is accurate, but also taking a look at whether or not we think we're missing some data. That can definitely happen. Did the, mass spectrometer run short? Did we not collect an entire sample? Those kinds of questions. So that's kind of interesting. Sorry, I just yelled. That's kind of interesting. The next thing that we can do is view our data sorted in order of chromosomes. And so one thing that we could do is sort our index, or we could use sort index to sort our data in order of chromosomes rather than in order of in order of number of SNPs. Okay, so let's see if we can do that. So I'm going to copy this and I'm going to paste it here. And there is one way of doing this. Well, I'm sure there's many ways of doing this, but one way that we could do this is using the sort index function. So I'm going to try this. I think what I want to do is I want to grab this column. I want to get all of the value counts, but then I want to sort by the index, which is this column, not by the value counts. Okay, so let's try that. That should be a dot. And then that I basically want to plot. So what we're doing here is we're grabbing the column, we're grabbing the counts, we're sorting by the index, and then we're plotting it. Let's see if that works. No, that doesn't work. What did I do wrong? Hang on. Let's delete this and make sure that this sorts properly. Make sure I didn't do anything funky there. Nope, see, that didn't work. Not supported between instances of string and integer. Oh, is it not sorting? Can we, oh, we can't sort by object, is maybe that why. Sort. I think we should be able to do that. I don't know why I'm getting an error there. I was not expecting this error. This is why I love live streaming with y'all though, because the whole point of these is to learn how to figure this out together. So if, oops, I just shook my, there we go. I shook my, my thing there and then it all went away. Okay, so I want to sort by index. Let's see, pandas. I just realized you can't see what I'm searching for. And if this doesn't work, I want to continue on and we can explore it further in Thursday's stream. But we'll go ahead and give this a quick debugging try. So I want to sort index, but I want to do it on the, so let's go ahead. Yeah, because this is, it's trying to sort, but it's, because it's an object instance, it's not letting me do that. Sort index. Sorted object. Yeah, but I want to sort index by, let's see if maybe there's a, I want to sort index even though it's an object, basically a string. Let's see. This person is just trying to compare. Oh, do we need to like, interesting. So this one is just saying to typecast it as an integer. We couldn't really do that because we do have an X and a Y. We could potentially change the X and Y to be like 23 and 24 as kind of just a representation. But let's see if they have one. Let's see if one person. Let's see if we can quickly find this. If we are trying to do a sort index. And if not, that would be my recommendation, which we're not going to get into right now. Yeah, I think we're just going to not get into that right now. But yes, I think basically, well, maybe doing a two-string. Yeah, I mean, I think basically what I would probably do is actually just convert 20 or X and Y to 23 and 24, just so then that way we can make it easier. But I would also ask my SME if that's a problem, just in case 23 and 24 are reserved for something else. Okay. Moving on. This is interesting in terms of the number of SNPs per chromosome, but something that might be important is whether or not these values are unique. Again, because our data is coming from the real world, sometimes we might be getting duplicate data, and sometimes we might be getting duplicate data that has, maybe the same the same SNP ID on a particular chromosome, but it has a different genotype, and then that might be kind of like an error. And so those types of things we want to start to look into. So let's go ahead and grab this again. But this time, instead of just plotting all of the value counts, what we're going to do is we want to specifically only grab the ones that are unique. So what we're grabbing here is the column chromosome. We're getting the value counts from that. And then before we plot, what we want to do is that returns, remember, that returns this list, right? So now what we're going to do is from this list, we're going to look at the chromosome column again and make sure that they're unique. Okay, so we're going to, from that list, look at that chromosome column again and make sure that we have unique values. All right. So it's hard to kind of tell, but we can see this time it does look like it actually sorted it for us. Did that sort actually work? Interesting. I'm actually curious. It looks like it sorted it for us. Genome. Oops. Chromosome. Nope, that's just the order of these anyways. Not sure. In any case, this is only grabbing the unique ones. And then we can, again, similarly graph that, right? So we can grab all of this information and we can add in. I'm a big proponent of, or I'm a big believer in copy and paste. It allows you to not make a lot of errors as long as you know exactly what you're copying and pasting. Okay, so we can start to see that there are some differences. So if we scroll up to this one, we can see, oh, again, they're just kind of sorted differently, I think. But this is now sorted and we're looking at just the unique values. So looking at all of the counts is interesting, but being able to group our data by a certain column will allow us to explore those particular features. So, for example, we can group our genome data by the chromosome column and grab the count for all of the other columns. So we can see here that we've got, and I mean, in this case, all of the other columns have the same amount. But for chromosome 1, this is the amount of rows that we have for chromosome 2. This is the amount of rows that we have for chromosome 3. This is the amount of rows we have, et cetera, et cetera. The benefit of grouping by different columns is we can start to get different counts. So for example, we can group by something else like genotype. Remember, genotype is A, AA, AC, AG, AT, et cetera, et cetera. So all of those different combinations. And then we can look at the count of chromosomes that have that particular genotype. So of the genotype AA, how many chromosomes do we have that are represented with that genotype? How many SNPs, how many chromosomes on, okay, sorry. From this genotype, AA, for example, let's take a look at that one. AA, we have 150,000 unique SNPs for that chromosome, for each chromosome. Looks like there's some questions. What are we trying to do with this genomic data? So eventually, we're trying to find, like, what scientists are trying to do is try to find anomalies. Exploring the data at first, this is just one way of looking at data science from the perspective of this type of data, which is what we're doing today. So something more interesting, for example, is if we started to look at the like a pivot table and then being able to show a stacked bar graph. So let me show that, because that'll start to make the data a lot clearer. So we can see how many SNPs. So there are nearly 150,000 SNPs with the genotype AA, but we don't know across which chromosomes or how many chromosomes that is. Like, is that all on chromosome 1, or are those across multiple chromosomes? And that's what we can explore now. So For example, we can create a grouped genome, which is basically our genome that is grouped by our genotype and then by our chromosome. And we can get the RSID, so basically count of all of the SNPs. And we can get our grouped genome, and we can rename our columns. In place is fine, like we can rename this particular data frame because genome is our data frame that's storing all of our original data. Grouped genome is our data frame that is storing this particular data. And we can take a look at that. Make sure we didn't make any errors. I highly recommend anytime you're doing things in Jupyter Notebooks, you do this kind of like gut check where you print out the step to make sure that that's what you expected. And then we can basically create a pivot table like you would inside of Excel to take a look at this. So we can have it be indexed on the genotype. And our columns can be the chromosomes. And our values can be the count of SNPs. Okay, so let's take a look at what that looks like. Pivot genome, group genome, pivot index, genotype, chromosomes count. Where am I? Chromosomes. What did I do wrong? Chromosomes. Am I spelling? Oh, it's chromosome, no S. Okay. So then basically we can see for each genotype, for each chromosome, the count of SNPs. And again, that's not like super useful in table format, so we can start to take a look at it here. So what we're seeing, oops, keep forgetting that E. What we're seeing here, and this is going to take just a second, there we go, is exactly what I was describing above. So we can still see that the genotype AA has about 150,000 SNPs. But we can actually see that those 150,000 SNPs are split up across these chromosomes. And we can specifically see that like chromosome 1 has a good chunk down here, or has like the most. Chromosome 2 has the next, and it kind of like gets less and less and less. So we can start to see the distribution of the SNPs across the chromosomes for particular genotypes. And again, I'm not a genetic scientist or bioinformatics scientist or a biologist, so I'm not entirely sure what this means, but the point of me showing you this today is to start to understand how you could start to have conversations between the person who understands the code and being able to kind of manipulate this data and show it in different ways without changing anything, and the person who understands the science. So you can take this graph to the scientist and say, this is what we have here. What are we seeing? Are we missing any kind of data? Are we seeing any anomalies? Is there somewhere you want me to dig into deeper? Should we look at this only looking at the ones that are on chromosome 19? Are we seeing just like this huge uptick in chromosome 19 with the genotype GG, et cetera, et cetera? Does that make a little bit more sense? Oh, we call them SNPs. Thank you, BritB92. I didn't know that you called them SNPs. Very useful. I appreciate it. Awesome. It may just be the order and colors, but we are seeing an actual count. And yeah, you're right. It does look like some of the colors are repeated. So we'd probably want to actually like label this and look at it deeper. But you can see that some of them, let's see, blue, orange, green, red, purple, blue, orange, green, red, purple. I think these are still all ordered in that way. Awesome. Okay, moving on. One thing that you can do that's useful is actually start playing around with the plot a little bit. Again, part of why we're doing this stream is to talk about the relationship between the person who's coding and the person who understands the science or any other field. Last week, I did a little bit of a stream on wine data, for example, and the composition of wine and the different alcohol count. And I don't even remember the other features of wine now. It wasn't because I need to be an expert in wine. I don't need to be an expert in genomics, but rather to be able to have a conversation between them. So one thing that you could do, and I'm just going to start because I'm running out of time, I'm going to start copying and pasting the code here, is actually create figures that can be used for reports that can allow for that conversation to happen. So for example, we can create a figure. Again, it's basically just what we did before. It's a stacked bar graph. But maybe we actually want to move that legend over so then that way we can see things a little bit better, right? So the location, we say we want it to be upper center, but then we want to move it over just a little bit and we can get a better visualization with it like this. All right, so we're running close on time, but a couple of things that might be useful is looking up, here, I'm going to grab a couple of these URLs. Some of the common, looking up the number of base pairs per chromosome. And so I just realized I'm not showing you this here. There you go. So, this is one of the URLs that are in the book, but in the notebook. But you can take a look at one particular chromosome, for example, chromosome 1, and you can start to look at the different health conditions related to the chromosomal changes. So specifically looking at neuroblastomas and noticing that chromosome 1 might have an anomaly where the deletion of these is what might cause or has some kind of connection to folks who have neuroblastoma. So about 25% of people with neuroblastoma have a deletion of these particular SNPs, if I said that right. So you can start to look at these base pairs and look at the different anomalies or conditions or things that might result on those particular chromosomes. And there's some more information on like mitochondrial anomalies and DNA mutations, and we kind of linked to that here as well. So we could, for example, start to explore just kind of like the differences between those base pairs and the genotypes that they're associated with, and kind of continue to explore similar types of data. The notebook goes into that a little bit more. And finally, one other thing I wanted to show you. There we go, open. The NIH does have this genome data viewer, which also allows us to kind of do this exploration via, looking at the chromosomes and, that did not understand that request. Interesting. Maybe it doesn't. But starting to take a look at some of the differences, browsing the genomes, and you can then generate more data sets to use in more exploration of this data. This is really just the beginning. Again, meant to just show a little bit of the data science. So, like folks had mentioned, they might be biologists who are interested in starting to explore data science and what that means and why. And really, this is just the starting point to how could you possibly get data that looks familiar to a biologist like this into a Jupyter Notebook to start to explore it in a way that makes sense programmatically. And that was really the purpose of today's stream. And on Thursday, we're going to dive a little bit deeper into this. And we're going to use different data sets. We're going to take a look at a mammals data set that includes, basically it documents the body weight in kilograms and the brain weight of mammals. And we're just going to start to evaluate the relationship between body weight and brain weight, and understand kind of what we can gather from that. The notebook on the GitHub repo has some suggestions for how to further explore. We kind of provided the quote unquote answers in there. So I didn't want you to get stuck at all. But what I invite you to do is to finish going through that notebook, check out the data viewer here on the NIH site, get some more data, some different data, start taking a look at it and see if you can find any anomalies. One of the things that we didn't get to today was you could actually I think we do have that. Did we show it here? I think it's right here. So if we were to group by chromosome versus grouping by genotype, we can see that we do actually have chromosome 21 twice. And so we might want to, for example, combine those rows so that we're looking at chromosome 21 all up and not splitting those. Again, that would be a conversation to have with the biologist. Why might that show up twice? Are there different anomalies that we might be looking into, etc.? All right. And yes, this has been recorded. I'm going to switch over to my face again. This has been recorded. Thank you, Megan, for putting all of the links in there for us. The GitHub repo has this notebook in there with a lot more explanation around it. The data set is in there as well. Again, it's not under the data folder. And this will be posted on YouTube. And as you start to explore, I highly encourage you to go to that YouTube and write in the comments anything that you might have learned. Or you can tweet at us. And yeah, again, tomorrow, I'm going to actually ask my husband to help me answer some of the questions as to, what is the relationship between someone who's a machine learning expert and someone who's a biologist, and how does something like Jupyter Notebooks and Python in general play a role in that relationship and being able to actually work on real world problems. The type of work that he's doing, again, is proteomics, which is slightly different. But essentially what they're doing is taking samples from just like our Earth and running those just kind of samples through mass spectrometers, identifying proteins that exist in those, and then seeing if those match any known proteins that might help different conditions or that might mimic different conditions or things like that, so that they can begin to develop therapeutics based on things that already exist here. It's kind of neat. Why do I use Python 3.8? It is, the currently stable version. I literally got this machine on like Thursday of last week, so I set it up, and that was the newest Python, so might as well install it. 3.7 should probably work. Definitely don't use 2. It's deprecated now. So great. If there are no more questions, I'll stick around in the chat for just a couple more minutes. But otherwise, I will see you all tomorrow on the Twitter. And I'll place in the Twitter here as well. Thank you, everyone, for joining us today. Thank you, Megan. Yes, if you wouldn't mind, we would really, really appreciate it if you could fill out that survey. There's an event code in the chat, and I'll also show it just now.